today's Life Sciences lesson. My name is Vedika Maraj, and this lesson is brought to you by the Gauteng Department of Education in collaboration with Sibona Discovery Center. If you have any life sciences related questions, you can WhatsApp us during the lesson on 063 508 1301. Please remember that when you send a message, do not send videos, voice notes, or photos. Just text your question and we'll see how best to answer you. Today's lesson is going to be revision paper 1, but we are going to be focusing on specific challenges that were identified in the preliminary examinations that you just wrote. So you need to listen very carefully. The first one that we found that was an issue in the prelim examination was the menstrual cycle. So let's just watch this quick little presentation. Let it start again. That is just a diagrammatic explanation of what is happening in the menstrual cycle, okay? I don't want to say anything about it because I want us to go into it with regards to when we're looking at the graph, okay? But what that basically showing, let me just go through it again. It's just showing us that the ovum is going to be released from the ovary into the fallopian tube, okay? And then it's going to travel down the fallopian tube into the uterus, and then it's going to be expelled. And as you can see, the red area is showing us the endometrium, which is being expelled, okay? So the menstrual cycle is divided into three different cycles. It's two cycles, sorry. It's the ovarian cycle and the uterine cycle. The menstrual cycle is the 28-day. What happens, the events that happen in a 28-day cycle. That is the menstrual cycle. But within the menstrual cycle, we have an ovarian cycle, which shows us the events occurring in the ovary, and we have the uterine cycle, which shows us the events that are happening in the uterus. And that is one of the main places where learners go wrong. They don't understand that. So if we have to look at it in general, the table that I've put here for you is basically to show you the major events that you need to understand about the menstrual cycle and you need to remember. The first one is that from day 1 to 7, we have menstruation. That is when the lining or the endometrium breaks down and is released. Okay? Between day 8 and day 13, estrogen is then stimulates the endometrium to become thicker, more glandular, and vascular. That's happening between day 8 and day 13. When we reach day 14, that is when we have ovulation. Day 14, we've got ovulation. Then we've got day 15 to day 22, when progesterone stimulates the endometrium to become even more thicker, more glandular and vascular to receive the fertilized ovum. Okay, let's just talk about that a little bit. What's the whole point of having this menstrual cycle? Okay, one of the major events that happen which are contributing to reproduction is the release of the ovum during a process called ovulation. So every month, there's the possibility for a female to fall pregnant, provided an ovum has been released and obviously sexual intercourse has occurred, and that the sperm can then have an opportunity to swim towards the ovum to fertilize it. Okay, so that's what basically happens. But in the absence of fertilization, that is when the ovum is then going to be expelled during menstruation because the ovum is not fertilized. Okay, that is the understanding of the menstrual cycle that we need to have, which we find that learners are not really grasping. Now, once you know those major events that are occurring, let's look at a deeper view about what's happening in the menstrual cycle, because you also need to understand it a little bit deeper, because when the examiner is going to ask you questions, they're not just going to ask you what happens on day 14, what happens between day 1 and 7. They are going to give you graphs, they're going to give you extracts, they're going to give you different things and ask you to apply your knowledge. So let's have a look at the 28-day cycle. Like I said, we have the ovarian cycle. That's what's happening in the ovary. So I'm just going to write here ovarian cycle to show you that that is that one. And here we have the uterine cycle. Right. So before we do that, I just want to show you something. It's just an added point about the endometrium. I want you to look at this. The uterine cycle. Yes, we're talking about the uterine cycle, but here, can you see we're using the word endometrium? That is very important. When you are talking about whatever is happening to that lining, let's use the word endometrium. That is the most correct word, and that is the preferred word that is expected by the examiners. So let's look at what's happening over here. Right, day 1 to day 7. As you can see at the bottom here, day 1 to 7 is saying that we have menstruation occurring. Okay, so why is menstruation occurring here? We also need to look at this graph from day 1 to 7, from the ovarian cycle, what is happening to what is happening to the endometrium. Okay, so we already identified menstruation is happening. Let's look at the ovarian cycle. What's happening here is that we have the development of the primary follicle. These are your ovarian follicles that are indicated here, right? And here we have the development of the ovarian follicle. That's what's happening here. And we can see here that our hormone levels are low. So let's have a look at our hormone levels that we have here. We have FSH and LH, and these are pituitary hormones. Which means that they are secreted by the pituitary gland. Then we've got estrogen and progesterone, which are your ovarian hormones, and they are secreted by the ovary. But where exactly are they secreted? We're going to talk about that. But the main thing I want you to understand here is that in day 1 to 7, we have the development of the primary follicle, and we have estrogen and progesterone levels low. Estrogen and progesterone are low here, and that's the reason why the endometrium is going to shed, because progesterone and estrogen are low. Then, once that has happened, from day 7, you will see here that the hormones are all increasing except for progesterone. Progesterone is the blue one, flying a little bit low, but the others are all increasing. Why? FSH is called follicle stimulating hormone. That one is going to be secreted by the pituitary gland, and its name explains exactly what it does. It stimulates the follicle to develop. So we've got the follicles developing into the Graafian follicle, and that Graafian follicle is then going to secrete estrogen. Okay, that's what that arrow is for, to show you that that Graafian follicle is going to secrete the estrogen. That's why it's pointing to this red line over here to show you that it's going to be doing that. And then, as you know, from day 8 to day 13, we said that estrogen stimulates the endometrium to become thicker, more glandular and vascular. That's what the estrogen is doing. Okay, now we come to day 14. What is the major hormone that's going to be involved here? We're going to have this increase in estrogen. We're going to have the surge of estrogen because the Graafian follicle is now big, it's now mature, and remember, it's carrying that over. Now it's going to be that surge of estrogen is now going to stimulate a peak in LH. We call it a peak. Okay, it's a peak in LH. The LH is going to peak at day 14, and that peak in LH is going to cause ovulation to occur, which is the release of the ovum from the ovary. Okay, that's going to be happening at day 14. That's like a major event. Then what's going to happen? From day 15, we now have progesterone. But where does progesterone come from? That Graafian follicle that is now empty. LH is now going to stimulate that Graafian follicle to become a corpus luteum. And that corpus luteum is going to secrete the progesterone. Can you see now the progesterone levels have increased? Okay? Then what's going to happen then is, as we are reaching day 28, now the body is realizing fertilization has not taken place. And there's no Graafian follicle. So if there's no Graafian follicle, that means estrogen is going to drop a bit, okay? Because where will estrogen be secreted? Estrogen starts dropping, the corpus luteum starts degenerating, or you can say it shrinks, or you can say it disintegrates. Those are the three words that we accept. The corpus luteum degenerates, disintegrates, or it shrinks, right? And then if it does that, it now cannot secrete progesterone anymore. So the progesterone levels are decreased. So as you can see over here, estrogen and progesterone have decreased. And that decrease in progesterone and estrogen causes day 1 to start again and menstruation to occur. The lining will now be shed because there is no progesterone, there is no estrogen, the levels are now low, so there's no build up of the endometrium, there's nothing keeping it there. Because remember, the other major function of progesterone is to maintain the thickness of the endometrium. So if there is not sufficient progesterone, then we cannot have the maintenance of the endometrium. And why do we need to maintain the endometrium? So for possible pregnancy. Now, what happens in an instance where we have fertilization taking place? How would the graph change? Now let's look at this graph. This is a different graph, right? But I want you to look at it. What do you think is looking different? I'm going to give you a clue. Look at the ovarian cycle. What is the difference between this ovarian cycle and the previous ovarian cycle? Another clue that you need to look at is the ovarian hormones. What's happening with the ovarian hormones? Okay? So, I've indicated it there for you. Look at the shape of the corpus luteum. The corpus luteum, it does not degenerate. It continues to secrete progesterone. So you see, here's day 14, ovulation occurs, and then this corpus luteum and this number 1, number 2, number 3, can you see that they stay the same size? They have not shrunk. So because of that, they are still able to produce progesterone, and that's what they do. They continue to secrete progesterone. Why? Because if fertilization occurs, what do we want to happen? We want that zygote to develop into a blastocyst, and then it must implant into the endometrium. The endometrium has to be thick, it needs to be glandular, it needs to be vascular in order to keep or hold that and to receive that blastocyst. So we need to have our progesterone levels high. So that is what this graph is showing you, that if you see a graph where the progesterone levels remain high after day 28, and we've got the corpus luteum that does not shrink after day 28, then we know that fertilization has occurred. This is just a flow chart summarizing everything that I said. We are not going to go into it a lot of detail, but the main thing I want you to look at over here is that what is FOLH? FOLH stands for follicle stimulating hormone, estrogen, LH, and progesterone. So you would read it in this order. The FSH causes the primary follicle to develop and secrete into a Graafian follicle to secrete the estrogen. And then we've got the LH that comes into play, which is going to cause ovulation to occur and stimulate the formation of the corpus luteum so that we can release progesterone. Okay, that is basically what is happening over here and what is being explained in this flow chart. Okay, it's a very nice one for you to use and be able to understand how all the hormones actually work together. So you can see here's FSH, here's your estrogen, here's your LH, and here's your progesterone. Now, the question that was in the prelim paper that was difficult for some learners. Right, let's have a look at it. The diagram below shows a part of the menstrual cycle in humans. The arrows represent the direction of blood flow. Right. Now remember what I said. I said that the estrogen gets secreted by the developing follicles, but the most estrogen from the Graafian follicle. So when it gets secreted, what is actually happening? Remember, it's a hormone. And hormones are traveled via the blood, in the blood. Okay, so I think a lot of the time where learners go wrong here is that they think that, okay, the hormone is being secreted, estrogen is being secreted by the Graafian follicle, it's just coming out of the ovary. But it doesn't. It goes via the blood. So we need to think about this. The arrow, they're showing us an arrow coming in. The arrow coming in, which hormones would be coming in to the ovary? That would be the pituitary gland hormones, which is FSH and LH, because the pituitary gland is in the brain. So from the brain, they need to get to the ovary. How do they do that? It's not like by diffusion or osmosis or something. It's going to travel via the bloodstream. So the blood vessel is now going to bring those hormones into the ovary. Now, which hormones are going to be leaving the ovary? That will be your estrogen and your progesterone, because like I said, the Graafian follicle will secrete the estrogen, the corpus luteum will secrete the progesterone, and via the blood it will go to where it needs to go. In this instance, it would be needing to go to the endometrium. Now let's look at the questions. Identify structure C. Okay, so structure C, you can see that the small little follicles, so those are our primary follicles. You cannot call them follicles, you cannot call them developing follicles, they are the primary follicles. Okay, name the cycle shown in the diagram above which forms part of the menstrual cycle. Now you see, this is where learners go wrong. Like I said, the cycle that forms part of the menstrual cycle. And what did I tell you they are? There are two. They are the ovarian cycle and the uterine cycle. Right. So this one is the ovarian cycle because they are showing us the events occurring in the ovary. Arrange the letters A to E in the correct sequence of their appearance in the cycle named in question 2. Okay. So in this one, we have to see what is happening from the beginning. So it must be in a correct sequence of appearance. So we would obviously start with the primary follicle, right? Which is this one. Then we would go to the B, which is the developing follicle. Then we've got to A, which is the Graafian follicle. And then we have E. First E, which is the corpus luteum. And then we have D, which is now the degenerated or the shrunk corpus luteum. Okay, let's see the answer. Yes, that is correct. It is in that order: C, B, A, E, and D. So in this instance, the question was 2 marks, okay? And how they wanted you to mark, how they want you to answer it is the exact same sequence. It's not 2 marks all or nothing. It was the first 3 gave 1 mark, and then the other 2 gave another mark. So say, for example, you had written C, B, E. Even though your C and B is correct, but you don't have an A, you cannot get that mark. Or what most learners did was, they said C, B, A, they got 1 mark. Then they said F. Firstly, F was not part of the instruction, right? Secondly, they're asking us to arrange the A to E in the correct sequence of their appearance. F is actually a process that is occurring now, or showing the ovum actually. But they wanted you to just do A to E. So it's very important that we read the instruction carefully. Otherwise, you lose a lot of marks unnecessarily when you really know the answer. Draw a labeled diagram of gamete F and identify it in the caption. Now, where learners went wrong here is that they didn't realize that gamete F is an ovum. So that's the first thing that we need to realize, is that this is an ovum and you need to be able to draw an ovum. And I don't know, it's very strange for me in a way because it's showing us their F and you can see that it's a round structure. At least if you had drawn a round structure and put a nucleus, you would be able to get marks, right? So let's have a look at the drawing, and I'm going to put the rubric as well because it's very important for you to see how do we mark a diagram. So here we have a jelly layer. Now I want you to understand something about this jelly layer. It is not a membrane. A cell membrane is your membrane, right? A jelly layer is not a membrane. You can only say jelly layer. So the jelly layer is that outer jelly-like, gel-like layer. Then you've got a nucleus on the inside. We've got a cell membrane, and then we've also got cytoplasm in the inside. So we've got the jelly layer, then you've got the cell membrane, then you've got the cytoplasm, and you have the nucleus inside. Remember, it is a haploid nucleus, right? Now let's look at the rubric. You get 1 mark for the drawing if it's the correct representation of an ovum. In other words, it must be single-celled, it must be round, and it must have a nucleus. If it doesn't have those things, then you can't get a mark for a drawing because then what is it? It's a circle. Okay? Some learners just drew a circle. Any correct labels as shown in the sketch above. Okay? So you get marks for that, 2 marks for the labels, and then structure identified as an ovum in the caption. Okay, so you need it because they said identify it in the caption. That was another place where learners went wrong. If they had asked you, draw a label diagram of gamete F, then as your caption you can say gamete F. and you could get a mark for caption. But here they told you identify it in the caption, which means that you needed to, there's a mark for that. That's what, and there's still one mark, but you needed to identify your drawing as an ovum in order to get the mark. A high concentration of which two reproductive hormones will be supplied by blood vessel X? Right, so blood vessel X, like I told you, that is the one that is coming in. Let's look there. It's the one that is coming in. So remember I told you those would be the pituitary gland hormones, and those are LH and FSH, right? I hope you got that right. Transported, and which hormones will be transported to other structures by blood vessel Y. So that is the hormone that will be leaving, and that will be estrogen and progesterone. Explain how the formation of structure D will lead to the start of the next menstrual cycle. Now let's look at D. D is now this degenerated, disintegrated, or shrunk corpus luteum. That means that, now just let's get the understanding before we go to explain. If it's going to shrink, there's going to be no progesterone. If there's no progesterone, the endometrium cannot be maintained. If the endometrium is not maintained, it's going to shed. Right, let's look at the answer. The degeneration of the corpus luteum, or the disintegration, or the shrinking, leads to a decrease in progesterone. The endometrium is no longer maintained, or you can say it's shed, or you can say menstruation occurs. Thus, FSH increases, causing the next cycle to start. Okay, they're asking you how would it lead to the next one. Now, I didn't mention this earlier, but I'm going to explain to you now. The other thing about fertilization, when fertilization occurs, we are not going to have FSH levels increasing, because once an ovum is fertilized, we don't want new follicles to develop, because we don't need another ovum, because fertilization has already occurred and the female will be pregnant. But in an instance when we have when menstruation has now occurred, remember the progesterone levels are now going to decrease, and when the progesterone levels decrease, it's going to stimulate FSH to increase again, so that the development of new follicles can occur, so that a new ovum can develop. So that a new ovum can develop. But when progesterone levels are high, it inhibits FSH. So because we don't want new follicles to be developed. And when our progesterone levels high, after day 28, it is when the female is pregnant or there's a fertilized ovum. Right. Another question. The graph below shows the levels of two hormones that are secreted by the pituitary gland during the menstrual cycle. State two functions of hormone B. So this is they're telling you now that it's two hormones secreted by the pituitary gland. So you must already know that we're talking about LH and FSH. And then the one, number B, how do you know what it is? You look at day 14, what is happening over there? We have this peak just before day 14, therefore we are looking at LH. What is the function of LH? It stimulates ovulation and stimulates the development of the corpus luteum. Explain why a female who is struggling to get pregnant may be given pills containing hormone A as a treatment. So if you're trying to fall pregnant, hormone A is going to be our FSH, okay? We need follicle stimulating hormone. Why? Because we need to encourage the development of, stimulate the development of the follicles so that we can have a possible ovum. So we have a high concentration of FSH to stimulate more follicles, therefore more ova will be produced. The key word here is high. When you're talking about hormones, you always need to say whether they are high, whether they are low, whether they are increasing, whether they are decreasing, or whether they are just staying the same. Because remember, these hormones are being secreted. So when you're explaining any event, you need to talk about what is happening more or less. Explain why a female who is struggling to get pregnant will have her levels of hormone B constantly monitored. A peak in hormone B will indicate that ovulation is about to happen, therefore an ovum will be available for fertilization. Okay, because remember we said the peak in LH is going to indicate that ovulation can occur, and if ovulation is going to occur, that means there's a possibility there will be an ovum that can be fertilized. Explain how the levels of hormone A on days 0 to 5 will differ in a pregnant female. Okay, so we look at day 0 to 5, how would they differ in a pregnant female? So normally day 0 to 5, we say menstruation occurs. But remember I said we need the endometrium if pregnancy occurs. So the levels will remain low because the high progesterone, so FSH will remain low because of the high progesterone levels, which will inhibit the pituitary gland from secreting the FSH. So when you do have a question like that, you need to explain that the progesterone level is high. First, you need to say hormone A is FSH. The progesterone level is high, which means it's going to inhibit FSH, and that's how you must explain it in a cause and effect manner. Describe the secretion of the ovarian hormones and their role in the menstrual cycle. Okay, we spoke about this one at length, so we're just going to go through it quickly. We have the Graafian follicle, which secretes estrogen and stimulates the thickening of the endometrium. We then have the corpus luteum, which secretes progesterone, which further increases the thickness of the endometrium. I just want you to understand that. Further increases, and this one stimulates the thickening. So estrogen is the building up and progesterone is the maintenance. It's very important that you understand that difference. Right, then we have the development of the zygote, implantation, and gestation. This is another part where we find that learners are struggling, the part that learners struggle with. So let's look at the ovarian cycle again. Here's the ovary. We can see ovulation has occurred over here, and then here we have an enlarged photo of fertilization occurring. And let's get the definition of fertilization correct. It is when we have the fusion of the nucleus of the haploid nucleus of the sperm fuses with the haploid nucleus of the ovum, okay, to produce a zygote, the diploid zygote. So the diploid zygote is then going to develop by mitosis. So I've put a little diagram over here to show you that we have the zygote, and then the zygote is going to then develop into mitosis is going to occur, right? The cell division is called mitosis that occurs here because that's the one that's going to be in charge of growth. So we're going to have the zygote that's going to develop into 2-cell structure, 4-cell, 8-cell, until we get to this solid mass over here, which is called a morula. It's a solid mass of cells, and then we've got the hollow mass of cells, which is called a blastocyst or a blastula. Okay, that blastocyst is now the structure over here. It's then going to move down into the endometrium, and then we're going to have implantation occurring. Let's look at this enlarged diagram over here. You can see here that this blastocyst is embedding itself into the endometrium, showing you something very important here. This is the uterine wall. This is the endometrium. When you are saying that the blastocyst is being implanted, you do not say that it's implanted into the uterine wall. It's implanted into the endometrium. Okay? Thereafter, it is then going to develop that embryo. Now, this is showing us a later stage where we have the fetus. Okay? But what's first going to happen is that we're going to have the outer layer here, which is called the chorion. This chorion is going to develop these chorionic villi. Okay? They're going to develop these chorionic villi. Those chorionic villi are going to extend into the endometrium. Then, together, these chorionic villi plus this endometrium is going to make up the placenta. I'll say that again. This is the endometrium, here's the chorionic villi. The chorionic villi, which is embedded from the chorion, you're going to develop chorionic villi. Those chorionic villi are going to now embed itself into the endometrium. The chorionic villi together with the endometrium is called the placenta, and that placenta is going to stay throughout gestation. Okay, and it's going to supply the fetus with nutrients. And the fetus's blood and the mother's blood don't mix. Why? But they come into close contact with each other. Why am I saying that? Because the endometrium belongs to the mother. The chorionic villi belongs to the fetus. So they are closely interlinked like this. But they are not mixing. The blood is not mixing. But diffusion is able to happen between them. Okay? Then we've just put a summary here for you of the different parts in gestation. Please make sure you understand them. What I like about this diagram is it shows you the uterine wall and it shows you here the endometrium. We must understand the difference between these two, because when you are using them, the learners are using the words interchangeably, and you lose a lot of marks because there's a difference between the wall and the endometrium. So you need to know what the placenta is. You need to know the function of the placenta. It's for mainly gaseous exchange, providing nutrients. You need to know about the chorionic villi. It's not called chorion villi. It's not called chronic villi. It is chorionic villi. It comes from the word chorion, which is the outermost extra embryonic membrane. The umbilical cord, you need to understand that there are umbilical arteries and an umbilical vein. An easy way to remember this is that we have 2 umbilical arteries in the umbilical cord and 1 umbilical vein. Then you need to know the amniotic fluid and the amnion and the functions of the amniotic fluid as well as the chorion. A very nice one-pager. You will find this in the DBE study guide. You will find this diagram, and really it's everything you need to know about gestation on one page. Let's go through a question. The diagram below represents the relationship between the blood system of the fetus and that of the mother. The arrows indicate the direction of blood flow in the blood vessels. Right, so this looks like a complicated diagram, but let's just have a closer look at it so we can see what is happening here. This structure here, the circle, is showing us the fetus. Okay. Then we've got a blood vessel of the fetus, which is blood vessel A, and you've got blood vessel B of the fetus. Okay, so these are your, this is basically the umbilical cord. We have one that is going to the fetus and the other one that's going from the fetus. And then here we have the placenta. So here we're talking about, this is the side of the mother. We've got blood vessels of the mother that are indicated over here and here. And then we've got label line Q and label line P. Okay? So what is structure P? Now we told you that those are those long finger-like projections that are called the chorionic villi. Then what is the extra embryonic membrane? The extra embryonic membrane, we spoke about those ones, and that is the chorion. Okay, so we have the chorion that develops the chorionic villi. Give 2 functions of the placenta. So the placenta has many functions, but you can name any of them in this particular question. It acts as a microfilter, because it prevents harmful substances from reaching the fetus, produces antibodies, secretes progesterone during pregnancy, which maintains the endometrium. Immunity is transferred from the mother to the fetus. Okay, so those are the functions. Those are the main functions, and you need to understand them. Learners get this wrong. Please make sure you know this. It acts as a microfilter, produces antibodies, secretes progesterone, and it's also there for immunity. And these are basically 3 of them here are protective functions. And they can ask you that sometimes. They can ask you, what is the protective function of the placenta? So you need to know that. Blood vessel A is a vein. So fortunately enough, they're telling you that it's a vein. Tabulate 2 differences between the composition of blood found in blood vessel A and found in blood vessel B. So in this instance, they were nice enough to tell us that A is a vein, so that you can, when you do your table, you can identify the vein, and then you can see the other one is the artery, and now you need to compare them. Please remember that when you draw a table, it's a full table, a full block with column headings, and to get your 1 mark for table, you need to put the column headings in. Right, so we have a high concentration of, in vein A, blood vessel A, sorry, which is the vein, we have a high concentration of nutrients. That's what's going to the fetus. From the placenta going to the fetus, what do we have? So you always think to yourself, what will the fetus need? Because that's what's going to come via the vein to the fetus. It's going to need nutrients. It's not going to need waste. So it's going to have a low concentration of waste, a low concentration of carbon dioxide, and a high concentration of oxygen. Now the artery will have the opposite. It will have a low concentration of nutrients, low concentration of oxygen, and high urea and high carbon dioxide, because these are the things that are coming from the fetus to the placenta. Okay, that is going to be held in that placenta now for the period of gestation. Explain one consequence for the fetus if blood vessel B becomes blocked, preventing blood flow. Okay, so they're saying that blood vessel B is going to become blocked. Here's blood vessel B, and we said that it was the artery. Okay, it's going to become blocked, and it's going to prevent blood flow. So this is not going to be happening because it's blood, so it's not going to be going there, right? the waste product and the nitrogenous waste is now going to accumulate. So in here, the CO2 is going to accumulate and the waste in the fetus, okay? And the fetus body is now going to be harmed, or the fetus is going to be harmed, or it's going to cause the death of the fetus because we don't have this being excreted, okay? Waste products become toxic in your body if they are going to be accumulated. If the blood of the mother and the blood of the fetus mix during pregnancy, it could lead to the death of the fetus. Describe how this could cause the death of the fetus. Okay, so now you've got to think to yourself, why do we not want the blood to mix? Firstly, if the mother has any diseases and the blood is going to mix, then that means that the fetus can get the disease. Secondly, blood groups. Remember that it's not necessarily that the blood group of the mother and the blood group of the fetus is going to be the same. And what do we know about blood groups? Okay, we know that we have different blood groups, and that they can actually you can develop antibodies in your body against them that can actually attack. So the only thing that you need to explain over here, you don't need to know the detail of it, about the rhesus factor and all of that. You might read about the rhesus factor in your textbooks, but you don't need to require that much knowledge about it anymore. You just need to understand that the harmful substances like bacteria or anything harmful could pass from the mother's blood to the blood of the fetus, or the blood types of the mother and the baby may not be compatible. That's as far as you need to explain it. Right, then we move on to the last section that I'm going to do before I hand on to my colleague that's going to be doing the rest of the session. We've got the homeostatic control of glucose. This question is almost always asked in one way or the other, whether it's in section A, whether it's in section B, but you are going to get the homeostatic control of blood glucose. Learners, you really need to understand this because you are going to see it, right? And we find that learners are really not doing well in it, but this diagram really explains it very nicely. Okay, so what we need to understand is, let's look at the narrative, because the narrative is actually going to explain before we have to look at the picture. After having a meal, your blood glucose levels will increase. Okay, so you always have glucose, but after having a meal, those blood glucose levels are going to increase because more glucose is absorbed from the small intestine into the blood. The pancreas detects an increase, because remember the food is now going to go through the small intestine. So now the glucose is going to go into the blood. The pancreas is now going to detect this increase in the glucose levels, and it's going to release the hormone insulin. And insulin is going to cause the excess glucose to be converted into glycogen. Insulin causes the excess glucose to be converted into glycogen. Glycogen is then stored in the liver and the skeletal muscles of the body. The body cells are also stimulated to take up glucose. This lowers the blood glucose level and returns it to normal, because now that excess glucose is going to be converted into glycogen stored in the liver and the skeletal muscles. But when the glucose levels drop, because the body cells are constantly using the glucose for cellular respiration, right? Glucose is needed. The body is using the glucose. Now your glucose levels are low. The pancreas will then release glucagon, which is a different hormone, which is going to convert that stored glycogen, or that glycogen that is sitting in the liver, or that glycogen that's sitting in the skeletal muscles. It's going to convert it into glucose. And this increases the blood glucose level and returns it to normal. Okay, so the diagram is just explaining that as well. It's showing us that when there's low blood sugar levels, we have the pancreas over here. Low blood sugar, we are going to get the glucagon, which is going to secrete the glycogen. The colors are showing you what's happening, right? We're looking at the turquoise one, this bluish color. We have the pancreas that's going to secrete glucagon. The glucagon is going to convert the glycogen into glucose, okay, which raises the blood glucose level. The high blood sugar causes insulin to be secreted. It stimulates glucose uptake from the blood, right? And it stimulates glucose into glycogen, and then it lowers the blood glucose levels. Okay, so this is the question that he had in the preliminary examination that learners did not do well in. The diagram below shows an endocrine gland involved in regulating blood glucose levels. It shows the hormone it releases when blood glucose levels are high and one of the target organs of the hormone. So they're telling us that this is showing us when blood glucose levels are high, the hormone that is released when blood glucose levels are high. It's very important that we read the stem. If you just look at the diagram, it is showing us hormone C. Let's look at the diagram. If we didn't read the stem, let's look at the diagram. We know that A is the liver. We know that B is the pancreas. Okay, if you didn't know, now you know, right? The pancreas is this, it looks a little bit like a tree-like structure, a long tree-like structure. Some people like to say that it looks like a grape, a vine of grapes. And then we've got this liver, which is this long, so you can see it's got different lobes. It's this big structure. But it's just telling us, showing us an arrow coming, a C, the arrow C coming out of the pancreas. But you know that the pancreas has two hormones. I just told you about them. I told you there's insulin and there's glucagon. So there are two hormones. So if you don't read the stem, you're not going to know which hormone that is. But because you read this, the hormone gets secreted when blood glucose levels are high, you must then know that this has to be insulin because the blood glucose levels are high. Identify each of the following organs A and B. We did that. It's the liver and the pancreas. Hormone C, we just said that it's insulin. And now they want to know what is a hormone. When they ask you what is a hormone or define a hormone and they put it in italics, they want you to basically give a definition. Okay, this is where your biological terms are very important and being able to write out a definition. It's a regulatory substance in the body that stimulates cells to bring about change. Or you can say you can also say it's a protein. It's a chemical messenger in the body. Explain how a lack of hormone C affects the blood glucose levels. So they're saying if you do not have enough insulin, what is going to happen to the glucose? So remember I told you that the insulin, it converts the excess glucose into glycogen. So if there's not enough insulin, obviously this is not going to happen. So how do we explain it? Because we need to explain it for 3 marks. We are going to say excess glucose cannot be converted to glycogen. Note there that I'm talking about excess because remember I told you glucose is there. It cannot be all the glucose that gets converted into glycogen. It's the excess that gets converted into glycogen. But in this instance, it cannot happen, right, in the liver. Thus, the glucose level in the blood remains above normal, right? So we said insulin is not there. It cannot convert the excess the excess glucose into glycogen in the liver. It's important that we mention the liver here. Okay, you need to know where it's happening. The glucose levels will then remain above normal, or you can say remains high. You cannot say the glucose, there is now glucose in the blood because there's always glucose in the blood. We are saying that the levels are going to remain high or above normal, and the person has diabetes. Okay? List 3 other hormones that can directly affect blood glucose. What other hormones affect blood glucose? These are thyroxine, okay? Because remember thyroxine is involved in metabolism. It's adrenaline, and it is glucagon, because adrenaline is also going to have an effect. Its effects, and then glucagon as well, because glucagon is the other hormone. But this was a little bit tricky for learners. The 2 parts where I found tricky of this question that learners did not understand is question 3 and question 4. But the main thing was that we are not using the correct words. We're not talking about excess. We are not talking about above normal. We are not talking about all the levels remaining high. Also, many learners did not realize that now this person would be diabetic. Okay? And then the other thing is that thyroxine, it has got to do with, it's also linked. You see, in life sciences, it's very important for us to understand that, remember, our human body is linked. Nothing works in silos. It's not like our digestion is now going to work, then my nervous system is going to work. Everything works together. So examiners love, in paper 1, if I can say something about paper 1, is that examiners like to combine systems. Okay, so just because you have the endocrine system doesn't mean we're not going to bring in the nervous system, because look at this now. When do we talk about adrenaline mostly? Yes, it's part of the endocrine. All 3 of these are part of endocrine, but they also have an nervous system implication. It's got homeostasis. So all of these systems work together. Right, that's all from me. We're going to take a break for a minute or 2, and then my colleague Nonhlanhla Mshali is going to be with you to take you through the rest of the lesson. Thank you.
of today's lesson, which is revision on paper 1, looking at the problematic questions or challenges that we have encountered, looking at our prelim question papers and the past question papers generally. So I'm going to be taking over and we're going to look at the eye and the ear, starting with the structure of the eye. So here again, remember when you do your diagrams and your structures, it is very important that you are able to label and you are able to provide functions. So we are going to look basically at the structure, not to go through everything, but just as a reminder, with what are the things that you need to look into when you're looking at the structure of the eye and the functions. And we always recommend that when you practice your diagrams, you get yourself a blank diagram, you put it in the middle of the page, sort of a mind map, and then you start putting in the lines, and then next to each line you put in the label and the function. And if possible, on the very same page, you then can do your accommodation, pupillary mechanism, and the eye defects, so that when you study, you go into your exam room, you only have this one page in front of you where you have summarized everything about the eye. So this is always a tip that I give to the grade 12 learners, that try and have your all notes of the parts of the different parts that we teach on the very same page, so that everything is in one place. So with the structure of the eye, remember it is the labels and the functions of the different parts of the eye. So we have the three layers, your sclera, which is the white inelastic part that covers the posterior of the eye, and this thing, it protects the eye. We also, the other function of the sclera is to maintain the shape of the eye. We also have the cornea in front, which is the transparent part of the sclera, and we all know that what this does, it allows the light to enter the eye. Because it is transparent and it is curved, and the fact that it is curved, it allows the refraction of what? Of light. We also have the middle layer, which is called the choroid. This one is the continuation of the ciliary body, the iris. It is dark in color, has that pigment because of the blood vessels in there, and its function is to absorb the light within the eye. And the fact that it has blood vessels, it provides nutrients and oxygen to the rest of the eye. And then the innermost layer, which is the retina, and here this is where we find the photoreceptors, and that is where then the stimuli, which is light, is converted into what? Into an impulse, and then it is transmitted into to the brain. So this is where the sight or the image is formed. Like I said, it helps to convert the light stimuli into an impulse. And then we also have this part of the eye here in front, which is called the aqueous humor. It is a cavity that is filled with a fluid called the aqueous humor. And then what is its function? Its function is that it also maintains the shape of the cornea, and it is clear in color so that it allows the light to pass through. Also supplies nutrients and oxygen to the eye. And then at the back of the lens, we have this part, which is filled with the vitreous humor, a jelly-like also fluid. Again, the function of this is to maintain the shape of the eyeball. It keeps it round, and also it roll in the refraction of light. And then there's more parts. We were just looking at the outside. And then if we go further into the different parts of the eye, we're looking at the ciliary body, which plays a role in accommodation, and it has the ciliary muscle. So this, what does is it allows the lens to alter or changes its shape. Then we have the suspensory ligaments, which are attached onto the ciliary body. So these are attached to the ciliary body, and the lens, again, the two, they help in altering the shape of the lens. And then we have this opening, which is called the pupil. This is what allows the light into the eye so that it goes onto the photoreceptors. And then we also have the iris. The iris being the muscles that surround the pupil. They also, the iris, its function or its role is to control the size of the pupil, making it big or small, depending on on where the exposure in terms of the dim and bright light. And then we also have the lens. The lens also plays a very major role in the refraction of light. So when it alters its shape so that you are able to see the objects that are close or the objects that are further away. And then there's also Let me just move quickly. And then we have this part of the eye, which is called the yellow spot or the fovea centralis. We all know that when your teachers teach you that this is where most of the photoreceptors are highly concentrated, especially the cones. They are found in there. And then it only has the cones, and it is the site for what? For vision. And then we have this part, which is called the blind spot. And here, the difference between the two is that here there are no photoreceptors. So there is no image or no vision that is formed there. And then we have this part of the eye, which is the optic nerve. So once the stimulus has been converted into an impulse, then it is taken by the optic nerve to the brain. Remember, you've just heard me now, I said to the brain. In paper 1, we are very strict when we mark. You cannot just say to the brain when you're talking about the impulses that are coming from the eye, but it is to the cerebrum. You must be specific and mention the part of the brain. So those are the things that we pick up when you mention. So it's not that you don't know, but each time you respond to questions, you need to be very specific as to which part of the brain is this impulse going to. So in this case, it is going to the cerebrum. So please take note of that. And then with the eye comes binocular vision. Remember, these type of questions, it's where you can find this in 1.2, where you're given a biological statement and then you must give a biological term, or you can be asked to define what binocular vision is. So here, just a reminder that, remember, we have there's two eyes, so this refers to the ability to focus on an object with both eyes, creating a single image. We know that you can see with one eye open and one eye closed, but if both of your eyes are opened, that it allows that ability to focus, and it creates a single clear image. So what is the importance or the significance of this binocular vision? So you must know this, because what does it do? It provides this depth of perception which enables us to walk up and down the stairs. So that balance when you walk, why you don't fall, it is because you're using your both eyes to focus on an object. It gives also a wide field of view, like you are able to see wider, and then it gives precise depth of perception, as in the size and the distance of the object that you are looking into. It also helps with the performance of skills such as catching, grasping, and locomotion. So these are things that you can practice. You can try doing things with one eye open, one eye closed, and then see when both eyes are open how these things come into play. So that is binocular vision. And then we also have accommodation. Those of you who have practiced on many question papers as you can, because we always tell you that when you study, you do the eye and then you take your question papers, you go through different questions just to see. You will see that you come across a lot of papers where there is question on accommodation. So what is this accommodation? Now, this is just a revision, remember. It is the ability of the eye to alter the shape of the lens to ensure that a clear image always falls onto the retina, whether the object is near or distant. So in other words, whether the object is far away or closer to you, at the end of the day, there should be a clear image that falls onto your retina, clear image that will be interpreted in the cerebrum so that you are able to see. So what happens here, we're looking at the changes in the shape of the lens. So the lens becomes less convex or it becomes more convex depending on the distance of the object that you are looking into. This is the table that you have seen so many times. We are going to repeat it again today to emphasize the importance of you being able to describe what happens when the question says describe accommodation, or it will not be as straightforward as I'm saying to you now when they say to you describe accommodation when you're looking at the object that is closer to you. That is just simple and straightforward. It's a level 1 type of question, but sometimes this question can come in higher order where you need to figure out whether the object is close or near before you can give the accounts of events. So with accommodation, when the object is less than 6 meters away, that is near vision. When it's more than 6 meters away, and then that is vision. So there's a mistake there. I hope you are able to see that. This is more than 6 meters away. This is less than 6 meters away. So here it is supposed to be more than 6 meters away. So there's a mistake there. Please fix it on your notes if you have this presentation already in front of you. And then with the near vision, we for sure most of you now are seeing this. So we are going to go through it again. We're looking at the parts that play a role in accommodation. So here it's the ciliary muscles that contract and then causing the sclera to be pulled a little bit forward. And when the sclera is pulled forward, it causes the tension in the ligaments to do what? To slacken. So they sort of become loose. And then the tension on the lens is what is decreased. Then the lens become more convex. So think of an elastic band that is being pulled like this. So if we say the ciliary muscles contract, the sclera is pulled forward, then the tension. So if you're pulling the elastic band away, pulling it like this, then the tension is higher. But if you let go and you bring it closer, so the tension is loosened and it decreases. Therefore, in this case, we say the lens become more convex. So it bulges out, it becomes more rounder, which are the terms that we discourage you to use, because the correct terms are more convex or less convex. So this is how the lens is going to look like. So to show that the tension has been released on the suspensory ligaments, they will look like this, and then the lens will be more convex. That is how then you are able to identify. So if you get a diagram where you must look at the eye to figure out whether this eye is focusing on the distant, on the near vision, you look at the tension of the suspensory ligaments, and then you look at the shape of the lens. If it is more flat and more bulging out, then that's an indication that this eye is focusing on the near vision and the lens is more convex. So then when the lens is more convex, it means even its refractive powers, they do what? They increase, so that a clear image is formed onto the retina. And then with far vision object that is more than 6 meters away, it is the opposite of the near vision. So here, the ciliary muscles, they relax. The sclera, which was pulled forward, now goes back to its normal state, because its normal state is being backward. So it is pulled backwards, and then when it does that, the suspensory ligaments are taut. They become tight, and the tension on the lens is increased. So then we said here the suspensory ligaments are taut like this, then the tension is less. Here, the suspensory ligaments will be pulled out, straight out like this, because they are being pulled backwards. And then the lens becomes what? Less convex. So it is not more bulgy, but it is thinner and sort of taut. Remember, we don't use those terms, but you use the terms which are less convex. So that is what we're looking into. And then the refractive powers then become less, so they decrease, and then the clear image is formed. So we are trying to make it easy for you. I know your teachers have taught you about this. So the mnemonic for the near vision, we say the ciliary muscles contract, suspensory ligaments slacken, the tension on the lens is decreased, and then the lens becomes what? More convex. With the far vision, again, ciliary muscles relax, the suspensory ligaments are pulled tight, taut, and the tension on the suspensory ligaments or on the lens is what? Is increased. Therefore, the lens become less convex. So remember the CC, SSTD, LM, and then the opposite on the far vision, which is CRSTTILL. So please practice and practice and practice until you understand it. So yeah, this is just a diagram to explain what I've said earlier. So if you look at the diagram without looking at the notes that are there, we can already tell that this eye, this part here, it's an indication that this eye is focusing on the what? It's on the object that is less than 6 meters away, because number one, the suspensory ligaments are slackening and the lens is more convex, whereas this one is looking at a distant object. So the object is distant here because the suspensory ligaments are pulled, they are tight, they are taut, and the lens is what is less convex. So this is what you are looking into. So if you get the diagrams like this, you should be able to tell the focus by just looking at the state of the suspensory ligaments and the shape of the lens. So remember, if it's less convex, it has less what? If it's less convex, then the refraction powers are also less. But if it's more convex, then it has more refractive powers. So it can bend more light. And then there's also pupillary mechanism. With pupillary mechanism, we are looking at when the iris controls the amount of light that enters the eye by controlling the size of the pupil. So that is pupillary mechanism. So with the eye, there's only two things that you must know. Okay, besides the structure, the two processes: accommodation, pupillary mechanism. And these are easy marks. Easy marks. That is why we're going through it again today, because we want to make sure that when you go into that room, you collect all the marks around the eye and the ear. Again here, we said the iris controls the amount of light that enters the eye, and because of the what? Of the muscles that are found where? In the iris. So in the iris, there's sacral muscles and there's radial muscles. So here the problem is, what we've picked up is that it's the confusion between the two. Remember, when we talk about accommodation, we are looking at the ciliary. It's the ciliary muscles. When we talk about pupillary mechanism, we are looking at the sacula. Sacula, because the pupil is there at the center, and then the sacula, they form a circle around the pupil. Hence, we call them what? Sacral muscles. And then the radial muscles, they form a radius. They extend from where the pupil is to the circumference. So the radius. So learners who are doing math, they will know about the radius and the circumference. Hence, we call them what? The radial muscles. So these are the muscles that we are talking about when we talk about pupillary mechanism. Two, sacral muscles, radial muscles. So again here, you need to compare between bright light and dim light. So you must know what happens when the eye is exposed to bright light. You must also know when the eye is exposed to dim light. So with the bright light, your explanation would be, when the eye is exposed to bright light, is that the sacral muscles, the ones that are forming the circle around the pupil, they do what? They contract. So sacral muscles contract. Here we have the CC there. And what happens to these ones that are extending from the center to the circumference, which are your radial muscles? Then the radial muscles do what? They relax. And when this happens, what happens to the pupil now? This opening that allows the light to come through. So the pupil does what? It constricts. Again, very important. It constricts. We are not saying it becomes smaller or it is reduced in size. We use proper biological term. It constricts, meaning yes, it becomes smaller, but we mark you correct if you write it constricts. So when it constricts, what does it do? It allows the less light to do what? To enter the eye. Now I'm going to go back. It's very important here. Here, remember, it is the sacral muscles of the iris. So very important when you explain. It is the sacral muscles of the iris, then you get your full marks. But if you're just going to write sacral muscles contract, which sacral muscles are you talking about? So it's sacral muscles of the iris contract, the radial muscles of the iris relax, then the pupil constricts, allowing less light, very important, or reducing the amount of light that enters the eye. because light is still going to enter the eye, but only less amount of it is going to enter the eye. So that is why this less is in capital letters, because then less light enters the eye. So please remember to write it in full when you explain. Then again, you have your C for circular muscles contract, radial muscles relax, pupil constrict, less light enters the eye. And then the diagram on the left is just showing, on the right shows you the difference. And then with the dim light, so if your eye is exposed to dim light at night, when you get up at night and it's dark, what happens? What are the changes that take place in your eye or in the pupil size? So here it is the opposite again. The circular muscles, now they do what? They relax. So the very same muscles that are forming a circle around the pupil, they will relax, and then the radial muscles are going to do what? Are going to contract. Then the pupil will dilate. Please, correct terms. They will dilate, and then the amount of light that enters the eye will increase. Then the more light is going to enter the eye. So here it's not reduced, but it will be increased. Then more light is going to enter the eye. So this is what we're looking into when we look at what? At pupillary mechanism. So remember, we're talking about the two muscles, circular muscles, radial muscles with pupillary mechanism. Whereas with accommodation, we are looking at the ciliary muscles, suspensory ligaments, and what? And the lens. So here, this table just summarizes what is the stimulus in both processes. It is the light. So here it is the distance. With pupillary mechanism, it is the changes in the light intensity in terms of bright light or dim light. And then the parts that are involved in accommodation, it's the lens, suspensory ligaments, and the ciliary muscles. Whereas in pupillary mechanism, we did talk about the pupil, the iris, and the muscles that are involved are here. Then what are the changes that take place in accommodation? It's the shape of the lens mainly that changes, that brings about the focus of the image on the retina. And then here, what changes is the diameter of the pupil, where it constricts or it dilates. Here the lens becomes less convex or it becomes more convex. And then what brings about the changes here is the suspensory ligaments and the ciliary muscles, whereas this side, it is the radial muscles and the circular muscles. So now let's look at some of the questions that were in the prelim paper and some questions from the old question papers. So you'll get diagrams, you will get diagrams in the paper, and then you must be able to study the diagram and answer the questions. So we are looking at the diagram that shows the conditions of the eyes for different light intensities. So clever learner, you already underline different light intensities when viewing the same object. So already you know, okay, here it's about pupillary mechanism, because we said the stimulus in pupillary mechanism, it's the different light intensities. So that's the question. So we have diagram 1, 2, and 3. Before we look at the questions, we always tell you, when you get a diagram, try and analyze the diagram and see if you can figure out what is going on. So already the statement has given you an idea that this is pupillary mechanism, and then the next thing you're going to look at your diagram. Now try to see what is going on. So if you look at diagram 3, the pupil is dilated. So we can conclude that this diagram is viewing an object, but in what kind of light? In dim light, because it is highly dilated. And then diagram 2, the pupil size is constricted. It's not the same size as this one. So here we can conclude that this eye is viewing at the object, but the light intensity, it is what? It is very bright. So that is what we are looking into. And then this one, we can say it is in between. It's not too dark, at the same time it's also not too light, so it is just in the middle. But these two, we can clearly tell that this is bright light and this is dim light. Okay, then the questions are as follows now. If we had enough time, I would have given you time to go through them, so then but we are going to work on them together. So then the question says, give the letter and the name. So again, when you read the questions, give the letter and the name, and you look at the marks. There's 2 marks there. So it means you get the mark for writing the letter of that part and the name of that part. So here, if you don't know the letter but you know the part, you get your 1 mark and you lose the other mark, but it's 2 marks. So we give the letter and the name of the part that contains muscles. So which part contains muscles that is labeled here? If you look at B, B is pointing at the iris, and we know that the iris has the radial muscles and the sacral muscles. So the answer for A should be B, iris. And then 2 says the part that is made up of tough white fibrous tissue. We said that is what? Is the sclera, which is inelastic. Remember, because it is made up of the white tough fibrous tissue. This tissue from grade 10, if you remember your tissues, it is inelastic, and the clue is that it is white. So the only white part of your eye is the sclera. So then let's look at the answer. So the answer for B should be A and sclera. So we said number 1, A is B, iris, and then 2 is A, sclera. So 1 mark for B and 1 mark for iris, and then for B 1 mark for A and 1 mark for sclera. Okay. And then in which diagram 1, 2, and 3 represents the eye of a person in a very bright area? We've already done that because we did analyze the diagram before we answered, and we know that that is what? Diagram 2, where the rods are stimulated the most. Remember, the rods are stimulated the most if the eye is exposed to dim light. So where the rods are stimulated the most, that will be diagram 3. Why? Because we said the pupil size is highly dilated, meaning this person is looking at the object, but in the room where they are, it is very dark, therefore the pupil size is highly dilated, but then the rods are very active because this person needs to have a clear image. So then for this answer, it is diagram 3. And then the next question is, which muscles are contracted in diagram 2? So diagram 2, we said it's focusing on the what? On the bright light. So bright light, the pupil size becomes smaller so that less light what enters the eye. So which muscles are contracted there? And the answer for that is that it's the sacral muscles. Sacral muscles, the ones that are around the pupil. And then which muscles are relaxed in diagram 3? So as you can see here, we always tell learners that you are not going to get, unfortunately with life sciences, you are not going to get questions that are straightforward. So again, the answer for 3B is the sacral muscles again. But what is the problem here is that the examiners are very tricky. They are tricking you because you would have written sacral muscles, and then again when they say relax, then if you write, some of you will write sacral muscles, but you'll start doubting yourself with table. They ask sacral muscles for A and sacral muscles again for B. So if you don't know your stories, then you start thinking, it can't be sacral, and then you cancel the other one, you change to radial. But if you know your stories from the table, what pupillary mechanism is, you will definitely see that. Remember, in dim light, sacral muscles relax. In bright light, sacral muscles do what? They contract. So just remember that when you respond to questions. And then again, we're looking at more questions so that you see how they are asked, more or less the same, and how you respond. Then again, we have the diagram that shows that represents the human eye. So here the question is first identify part F. So we always say to you, when you get a diagram, you put the labels. So you are the learner, the paper is in front of you. Before you could answer, you just look at your labels. Okay, A is pointing at the vitreous humor. B is pointing at the yellow spot. C is pointing at the blind spot. D is pointing at the lens. E is the cornea, and F is the iris. So now you know all the parts that are indicated there. So what is part F? Part F is the iris, this part that has the radial and the sacral muscles. So part F is the iris. And then second question is, state 2 functions of fluid A. So here, Problem is, if you don't know your labels, you've already lost marks, because if you don't know what part F is, you won't know that this is iris. They say here, name the two functions of fluid A. If you don't know what fluid A is, you won't be able to state the two functions and you would have lost marks. But you know that this fluid is the vitreous humor, the jelly-like one that is found, said at the back. We said it maintains the shape of the eye, it nourishes the eye when we were looking at the structures, at the functions in the beginning. We also said it does play a role in the refraction of the light. It also prevents the parts of the eyes from drying out. So desiccation as well, that's what it does. So let's look at the functions that are written here. It helps to maintain the shape, the eyeball, keeping its shape. It's because of the vitreous humor. Plays a role in the refraction of light, allows the transmission of light. What else it does? Prevent desiccation of the structure of the eye that is drying out. It holds the retina in position, which is this inner layer that has the photoreceptors. It also nourishes what? The eye. So that is the function of the vitreous humor. So it's many functions, but they only wanted two. So any of these functions could have been given you marks. Then describe the structural difference between area B and area C. So area C, we said C is the blind spot. So this is blind spot. And then we said area B, this is the yellow spot. So this is yellow spot, this is a blind spot. From what you were told, we know that here there's no image that falls onto here because there are no photoreceptors. Whereas in B, which is the yellow spot, that is where the clearest image in your eye is formed, because here it is filled with photoreceptors, mainly only the cones. So the structural difference. So the question says the structural difference. We are not asked about the function. So structurally, how do these two differ? And your answer would be structurally, B has no photoreceptors, or yes, no photoreceptors here, and then C, and then B has a high concentration of photoreceptors, which are what? Which are the cones. So let's look at the answer. So area B contains a high concentration of photoreceptors, which are the cones, and area C contains no photoreceptors. So it has no rods and cones. There's nothing on area C. That is why there is no image that is formed there. And then the last question here is, describe the process of accommodation that takes place when the object is less than 6 meters away from the eye. So what are you expected to describe here? You are expected to describe near vision, because the object is less than 6 meters away from the eye. And remember, with the describe questions, you are just asked to give a series of events that take place in this process. So in accommodation, when you're looking at the object that is closer to you, step by step in that logical manner for you to get the 6 marks. So exactly as it is from your exam guideline and from your textbook. So looking at the object that is closer, we said ciliary muscles, they do what? They contract. We said the suspensory ligaments, they slacken, they become loose. We said the length, tension on the lens is what is decreased, therefore the lens becomes what? More convex, and when it becomes more convex, then the refractive powers are also increased, so it becomes more refractive. So ciliary muscles contract, ciliary body moves closer, and then the suspensory ligaments, let me just go back, ciliary muscles contract, ciliary body moves closer, suspensory ligaments, they slacken, tension on the lens is decreased, becomes more convex, and the light rays are refracted more to focus the light onto where? Onto the retina. And that is exactly how you explain it, and then you get your 6 out of 6 marks. So you just describe, no explanation, nothing, you are describing. Then the other question about the eye is again, looking at the structure. This is our last question on the eye before we move on to the ear. Here, we're looking at the structure again of the eye, but now the story is more about the nocturnal animals. So nocturnal animals are those that are active at night and they sleep during the day. The bats, for example, the owls, they are mostly active during the night, and during the day they are inactive. So again, here, the different parts, Usti is pointing in the middle, in the innermost layer of the eye. So C is the retina of the eye, and then A is pointing at the lens, B is pointing at the pupil, and then the iris is labeled. So here, nocturnal animals sleep during the day and are active at night. So here the question says, explain. Now this is different from describing, because describing, I said, you just provide the events that are taking place in the process. So here you explain how. Remember, where they explain, you always say it is a two-part question. It's something that will cause something. It's a cause and effect. This happens. When this happens, then this is going to be the result. So each time you answer and explain question, this happens, and then when this happens, this is going to bring about this change, or this will be the result. It's a two-part question. So if your answer only tells us about one thing and there's no effect of what you've started with, then it's incomplete. Or you only tell us about the effect and there's no cause, your answer is also incomplete. You don't get your 2 full marks. So here they say, explain how part B. What is part B? It's the pupil. How part B of these animals that are active at night will differ from the ones that are found in the animals that are active during the day. So you know, when from pupillary mechanism, that in dim light, your pupils are dilated. They are dilated so that more light can do what? Can enter the eye. So part B, we said it's the pupil. So how will it differ? Part B of nocturnal animals will be dilated, more dilated. Why is it dilated? So that more light can enter the eye. So I've spoken about the pupil being dilated, the cause, and the effect will be that more light enters the eye, because they function at night, they need to see clearly. So the cause, the effect. And then part C of nocturnal animals will differ from that, the ones that are found active during the day. So what is part C? Part C is the retina. The retina, remember, has high concentration of what? Of photoreceptors. But we have the cones and the rods. And the cones, they assist us in seeing the bright colors. They play a major role when we're looking at the bright colors, whereas the rods also play a major role if we're looking at the dark colors, black and white sort of. And then, so here, C, nocturnal animals, how will they differ? How will C of nocturnal animals differ from the animals that are active during the day? So because they're active during the night, they need to be able to see in the dark. Then it means their photoreceptors will have more rods, so that it allows them again to be able to see and to form that clear image that will be then transmitted onto the cerebrum. So let's look at the answers. We said the first one can dilate more to allow more light to enter the eye. The second one has more rods, enabling them to see in dim light. So your cause, your effect, your cause, your effect, your 2 marks. Then the last question says, describe how the iris controls the amount of light entering the eye when a person is exposed to bright light. This is pupillary mechanism, and the person is exposed to bright light. So when you describe, you just give the series of events, the radial muscle, does what, and then the sacral muscles will do this, therefore the pupil will constrict. When it constricts, less light is going to enter the eye. So sacral muscles relax, radial muscles contract, pupil constricts, less light. Again, radial muscles of the iris, the sacral muscles of the iris. So we are not going to look into this question, but please, you have been provided with a worksheet. Your teachers have the worksheet with all these questions that we have included here. So take those questions and go through them. It's all the questions that I'm skipping now because of the time. And then while you study, do not forget the eye defects. These questions are also very important. The defects, your people that are short-sightedness, the eye defects can be short-sightedness. If you can only see objects that are nearby, and then long-sightedness, distant objects can be seen clearly, astigmatism and cataract. So this table summarizes what are the defects. Again, you do have this presentation, so go through that. Is the summary of everything that you need to know. What is the nature of the defect and how can it be corrected? Because they will ask you, you will be asked. There are questions where examiners will ask you, how can one correct astigmatism? So that's when you come in to say, if they wear glasses with the lenses that are shaped to correct the distortion, or the contact lenses that are done. Or you can even say laser surgery can be performed so that this is what is corrected. Again, because of the time, we'll move faster out of this. And then the next part is the ear. Again, here, simple and straightforward. Like I said earlier, when you start your blank diagram, you put in all the labels and the functions. Just remember that the three regions, the outer ear, the green one, the middle ear, which is represented in red, and the inner ear, which is represented in pink. So these are all the things that you need to know. And then with the ear, the second slide just shows what are the different parts and what are their functions. So again, you go through this on your own, you read the different parts and the different functions. This is just a summary. Already you have all these notes in your book somewhere, but you're looking at the summary. So here I just want to, one thing about the middle ear and how it amplifies the sound, because then the teachers have explained this to you, how the middle ear amplifies the sounds. Remember, the middle ear is separated by, there's a tympanic membrane that separates the outer from the middle ear, and then there's another small window that separates the inner ear from these ones. So we have the larger tympanic membrane and the smaller oval window. So this side we have the tympanic membrane, and then this side we have the oval window, which is small. When you look at it, it looks like a trumpet, and we know the trumpet, when you blow onto it this side, there's this sound, more sound that is coming out. So also the sizes of the ossicles that decrease from being the largest to the smallest as you go towards the inner ear. So this arrangement, what does it do? It helps to amplify the sound. So it is the way they are arranged, from the larger tympanic membrane, the sizes of the ossicles that become smaller and smaller up until we get to the small oval window, and that arrangement is the one that makes the sound even more. It amplifies the sound. Then the parts, hearing, we always say that this is the easiest because the movement of the sound waves. You just need to remember that we start with the sound waves that are directed and collected by the pinna, and then they are directed where? All of these is sound waves. They are directed to the tympanic membrane. So when the sound wave beat against the tympanic membrane starts to do what? To vibrate. So from the tympanic membrane to the oval window, we now talk about vibrations. So the stimuli is sound waves, but once it gets to the tympanic membrane, it changes to vibrations. So those vibrations are transmitted through the ossicles to the oval window, and then once it gets into the inner ear has cavities that are filled with fluid. So the vibrations then become what? They become waves, fluid waves or pressure waves. So we have sound waves that are converted to vibrations that finally become fluid waves or pressure waves. Then these are the waves that are then converted into an impulse, which is then transmitted to the cerebrum if it's hearing, or to the cerebellum if we are looking at the balance and posture. So again, here we're looking at how does the ear regulate balance. So how does it regulate balance? What you need to remember here is the two things: the change in the direction and speed, and the change in the position of the head. So you must know the change, know the receptors and where they are found. So with the direction and speed, the receptors are the crista, and where are they found? They are found in the ampulla of the semicircular canals. So the inner ear, remember, it's made up of this part, which is the semicircular canals, and this part, which is called the cochlea. So the semicircular canals are these three canals here, call them semicircular. And then at the bottom, these parts are called the ampulla, and then that is where the receptors which detect the change in the direction and speed are found. And then with the position of the head in relation to gravity, the receptors that are responsible for that are the macula, where this part of the inner ear, which is called the sulcus and the ventriculus. So these are found. So both of them, any changes in this will stimulate the fluids, and then the fluid will the movement in the fluid will change will be stimulated, or the receptors will be stimulated, and they will be converted to what? The stimuli will be converted into an impulse, and then this impulse from these two parts of the ear is directed to the cerebellum of the ear and not the cerebrum. So those are the two things that you need to know. Again, I said, you do not just say to the brain, but you must be specific as to which part of the brain does the impulse go to. So let's just quickly look at this activity because we are running out of time. This will be the last. There's more that are here. You can do those ones on your own. So this diagram represents the part of the human ear. Again, you try and put labels. A is the tympanic membrane. B is the round window. D. B are the semicircular canals, and C is the cochlea. So identify part C. This is part C, the coiled part of the ear. It's called the cochlea. State one function of part D, or part D is the round window. Remember, once the waves are in the inner ear, if they come here, this is where they are stopped. It sort of reduces the pressure waves so that they don't come back and cause the echo. So that is what happens with the part D. And then the receptors found in part C, the receptors here, the organ of Corti, that is for hearing. Those are that's the organ of Corti. So these ones are stimulated and the messages are sent to the cerebrum, and then that's how hearing takes place. So the receptors here, they convert the stimuli or pressure waves into impulses. So these are then the answers: absorbs excess pressure waves. Like I said, this is the round window, and the one at the top is the oval window. So the last question here is explain why the buildup of earwax at part A, if there's buildup, may result in the temporary loss. So remember we said the tympanic membrane, when it gets hit by the sound waves, it does what? It vibrates. So imagine if there's buildup of wax. When it accumulates, it becomes hard. So it means the tympanic membrane will harden and it's not going to vibrate. Therefore, sound waves will not be transmitted onto the middle ear. Therefore, there might be no hearing. They will be impacted or to be reduced hearing. So part A, tympanic membrane, will not be able to vibrate or vibrate freely. Therefore, less vibrations will be carried to the middle ear. I like this question again because it tells you again what we always preach, cause and effect. So explain it will not be able to vibrate, and if it cannot vibrate, what happens? Less vibrations will be carried to the middle ear or the ossicles. So then we have come to the end of today's lesson. You do have the worksheet that has got all the other questions that we're gonna go through today. Unfortunately, we have run out of time, and please do tune in next week for paper 2 revision, which is on the 26th of October. Wishing you all the best for the rest of the year. Thank you.